Tritoplura.
Oh, thank you. Well, we have made, vertically, we have gone 45 meters.
Copy that now.
And we are an hour into the dive.
Negative. Come on in video. Quick zoom on this.
There's a tiny sponge. Have we seen this one before, Shirley?
Probably.
Should we move on?
Yes. Probably should move on. That's good. We can just get a good snap zoom of it. Sometimes you hear us saying get a snap zoom, so that gives us the opportunity to go in, take some quick video imagery of some of the things that we've seen earlier on this dive. And although I'm not sure we've seen this one earlier on this dive. And then we have the option as we zoom in and take a look at it. If any of our scientists want to take a closer look or stay longer, we can do that. Oh, look at that. Nice. Look at that slope.
I'd probably have it
yeah,
75.
this slope is pretty steep.
That sponge was actually a dema sponge. It's
really?
Stellodorix, that weird stalked one that we've seen in the Northwest Hawaiian Islands. So that was a nice one to zoom in on. I think it's the first time we recorded that in the Marianas.
Great. You'll go ahead and type that into the chat log then, right?
Just put it in.
Great. Thank you. Oh, we have something candelabra shaped coming up here.
We do indeed.
Oh, let's see, what is that?
Come on in video.
This one's.
Okay, not so much candelabra shaped as I thought when I first looked at it.
True.
Partial.
Alright,
Good in or out?
I'm stable, so you can come in.
Okay.
Are those brownish worm-looking things on the rock aplecopherans?
Yeah, I was just noting those wormy tubes.
Shirley, I think that's a good bet.
I'm getting better now that we're almost finished this leg. Okay, so what we're looking at here is another potentially a Candidella, possibly Narella, but Candidella, we're thinking. And then, pilot, when we're finished there, can you go just over to the right a little bit to 2 or 3 brownish worm-like animals? So a couple, 3 days ago, they were really tiny, right next to the coral. We saw...
And Shirley, while we go over there, I'll just note, sorry.
Yeah, go ahead.
Note that this coral could likely be in the same genus as that unbranched whip one, which just shows how much morphological variation there can be within a single genus of coral. And I think after we look at these possible aplecopherans, just to the left of this coral, there's an unbranched whip. You wouldn't have to move, just look left.
Thanks, Scott. Yeah, these are looking like they're aplecopherans. We collected a whip coral with an aplecopheran coiled around its base a few days ago. These are predators, and on that particular whip coral, that aplecopheran had managed to kind of just move right up the stalk and eat the polyps. So it was on that particular dive, we saw quite a few corals that were kind of say ratty looking.
I think that'll be good.
Looked like they had been preyed upon by this worm-like gastropod.
Yes.
Bridge, this is Nav.
Yeah, worm-like mollusk. Yeah, so that's a very cool observation. I suspect they are on the lookout for a coral to climb on and start eating, either a recruit or something smaller.
Yeah, sorry, not a gastropod. Worm-like mollusk. It does look like an amphipod stick, doesn't it? But it looks like it's an amphipod stick that might have been formed at the tip of a sponge stalk.
Exactly. At first I was going to say, hey, is this a sponge stalk or a cladorizid? But then I saw those amphipods on the stick above.
I know, and it's got at least one barnacle on it and maybe two, or could that be a...
Looks like a gastropod below the
looks like a
thing
limp.
with
It looks
the,
like a limpet almost, but I'm not sure.
The thing with the tentacles right below it? Does
Yeah,
it
so
have
there's
tentacles?
a barnacle and then the one right below it. Yeah, I'm not sure, Patty.
The nice thing to note here with the amphipods showing with the barnacle is that both of those are crustaceans. So even though that barnacle looks like something very weird because it's in that shell, it's actually a crustacean, and those are its legs that are sticking out of the shell. So it's basically much like that amphipod, but upside down, sticking its legs out of a shell that it creates.
And it looks like there's another tube that's attached to this sponge spicule stalk as well.
Covered with sediment.
But it looks like there are
Agglutinated.
tentacles sticking. Quite frankly, it looks like there are tentacles coming out of both ends of that.
Yeah, well, those are amphipods. So those are the antennae of amphipods, and there are two of them, one on the top and one on the bottom. So
wow.
We're getting to see a lot of the different ways that these crustaceans can make their lives and build their little houses. So these two are building tubes on the side of a sponge, and the other one seems to build a mud stick at the top of the sponge.
Right. Very interesting. So Tara Harmer-Luke, you can use this in your deep sea biology class as well. Les Watling from University of Hawaii was mentioning the other day that this is like, rolling invertebrate zoology classes where we are, getting examples at, in real time, in play, seeing these animals, in their natural environment and, exhibiting the behaviors that, in many cases, we've never seen before.
Is that a sea pen? Come in.
Does look like it to me.
Have we seen this before, Shirley?
I'm not sure if we've seen it on this dive.
Maybe a frog. I don't know what that is. Come on in.
Yeah, this is the backside of a bathypathes.
Okay.
But it's not clear whether it's the same one that we were looking at earlier. I assume Tina will be able to tell us that.
Can we look right at the base of that as well, please, pilot? Thank you.
There's the base.
Yeah, the angle we're looking at this black coral, we won't be able to see the worm. You have to see it from the front.
Yeah.
So I wouldn't spend too much time looking for a polychaete on this one.
Yeah,
Roger that. Thank you.
Yeah, we have to move
Roger that.
anyway.
Thank you.
I did see a large colony coming up to the left.
That would be perfect.
So if you want to partially deploy my upper swing arms a little bit, Sean,
Yeah.
just like
Doing them
20 degrees out.
from their starting point.
Yep.
Copy that.
Is this sponge of interest? Let's do a partial on this video.
We can do a snap zoom on that, please.
Partial.
Chris, you want to look at this more carefully than a snap zoom?
Yeah, you can come in tighter.
No, that's good. This is what we call Corbitellinae new genus. With all those projections out of the surface, it's a soft sponge. We've collected presumably the same thing in the northwest Hawaiian Islands. So it's a pretty distinctive sponge.
Has it ever been reported from the Marianas?
I doubt it, just because there hasn't been that much work in these depths in the Marianas.
Great. So
This type of sponge exists down here in 1 to maybe 3,000 meter range, at least in northwest Hawaiian Islands, and I just don't think that many surveys have been done here on sort of these colder seamounts, if you will, in the Marianas.
So we can add this to our list of new records of occurrence of these animals in the Mariana Trench
Let me know when you're good.
area.
We're clear.
Come on up. I gotta push ahead. I'm underneath you.
Yep, that's 80 on my cam.
Okay, we're seeing some more stalks
Nice. That's what I wanted, Sean, on the lights.
of dead sponges.
It is
The
nice.
stalks are heavily
Yeah, so
encrusted
when
with
you're up on a
manganese,
slope,
as are all
you
of
don't
these
need
rocks.
more light right in front of you
Yeah,
because it's reflecting
reflected
back in
right,
your
yeah.
face. So if you extend out your lights a little bit, you extend your light pool out, so it helps you find things, but it also gets some of the light out of your face.
yeah.
Quick zoom here. Is this just sediment, or is that a
Some
trail?
kind of trail or some kind of trail. Or it could be a tube, not sure.
These look like some kind of tube to me, formed from sediment glued together, maybe polychaetes.
All right, come on out. Get a scoop.
I think I can see a little ethopod down.
So, Sean, next time when you sit co-pilot,
Yes.
you should take a look at where the lights are and see if they need adjustment.
Okay.
I can't believe it.
You can't believe what?
Okay, pilots, looks like she was around.
We've made 17 meters vertically. That's it.
Well, we are on, we have made it through, this isn't, yeah, 800 meter. No argument.
Just
Yeah.
no arguments.
Just come in on this real quick. They may have already seen this.
Quick snap zooms, please.
Yep. Right, come in tight.
Great, thank you.
Okay, when you're good, video. Just come out. Quick zoom on this guy.
Okay, and that also looks like it may be the remains of a dead hexactinellid sponge. It does have some either anemones or zoanthids on it. That's great. That's great. Thank you.
When you're good, video. Are we getting enough time on those, Roland?
Tend to want, if we want to image it fully, it would be 10 to 15 seconds. I think we're a little shy. But
Okay. Yeah, so
don't know.
if you need a couple more seconds, and I say, you know, when you're good, then you can hold it that extra few seconds.
Okay, copy that.
Yeah, copy. Absolutely. Yeah, absolutely. I'm not setting down for any of these. I'm just pirouetting around, and we're keeping the ship on a steady move. Correct, Dad?
Correct.
So
Now being 4 minutes here.
my only concern is if we spend too much time zooming in on stuff that I have to zoom by, something that might be interesting to keep up with the ship. Come on,
Roger
video.
that, pilot.
Partial.
This may be a bamboo coral here.
The one to the upper right, not the one we're looking at
Nope.
right now.
This guy
Oh, the upper left or upper right? The one in this long whip, Scott, is that the one you're talking about?
Well, I don't know where you are right now. I'm looking at an Amphimastis, a pink sort of mushroom coral.
Yes.
A cup coral next to it.
Yes.
And then we zoom back on the upper left. Now it's come to the center of my screen.
That's a node.
There's
right
definitely a Chrysogorgia octocoral on the left side of the rock. And yes, that is a bamboo coral. Thank you. That's long enough to get a view of it. Good sclerite needles there, so that's good.
Great.
And yes, that is a bamboo coral. Thank you. That's long enough to get a view of it. Good sclerite needles there, so that's good.
Great. Thank you. Thanks.
And then I think I saw just to the background left of the bamboo coral whip, there is, I believe, a new kind of black coral, possibly an umbellapathes.
Okay, there may be a new kind of black coral.
Wow.
Okay, we'll take a look at that. I'm not. Are you talking about the thing that's on the stalk?
Yeah, the tip of that stalk, it kind of looks like a crinoid.
Yeah. Yes. Yeah, it does.
come on in.
Okay, we'll take a
I
look
think
at
it's
that.
actually a black coral.
Ooh, look at
Oh,
that. Look at that. Wow. Good eye, Scott.
Ev, how's that move coming?
Partial.
Now
Let me
we've been
just get my
stationed now
toe
5
down.
minutes.
Yes, that is definitely
Okay.
a black coral.
Yeah.
First
you do
we've
same
seen
thing.
at this dive. I think we may have seen one earlier. And Tina will weigh in. Yes, she has said it's the genus Umbellapathes. So nice to see that. Thank you. We
Yeah.
can move on.
Then, yeah, please.
Copy that.
We're good.
0 meters, 120 and 0.2 knots.
Well, what I'm trying to do, Nav, is get a little ahead if we, in case we have
Watch
to take
the
Pilot,
pilot.
there's something
Copy that.
floating. I don't know.
That's above me.
Oh, okay, never
Bridge.
mind. I
This is
thought it
Nav.
might float right in front.
Yeah, I see it. Watch lead pilot.
Go ahead.
Pilot. Yes, thank
Yes.
you. Just wonder if you could just
bearing
pause for
120,
just 2 seconds on the
80.2
rock
knots.
all of this is sitting on. I think it looks like it has less manganese on it, maybe a loose boulder.
Okay, copy.
Do you want us to cancel that move? Copy.
So this big rock.
Right. Yeah, thank you. That's good. I appreciate it. Thank you. Yeah, I'm interested in this rock because it's much more angular on the surface, you know, where all the animals are. And I'm guessing it may be tailless from the steeper slope that we're approaching as we go toward waypoint 3.
Go ahead, Bridge.
And
We talk about 10
yep,
degrees
it's
or
teetering on the
how much you
surface.
think?
So there's a
There are a
Yeah, it
lot
looks like
of
there's a
animals.
holosaurian maybe over on the right-hand side. Yeah, a
Right.
lot of stuff here.
also a cup coral
Copy that.
center
I'll just
on
confirm
the
with
lasers.
the pilots.
Come on in on the holothurian.
A pink-headed
Okay,
guy.
thanks for the info, Bridge. Bridge is bringing the ship around 30 degrees to starboard. Based on the center position, he said it shouldn't affect us.
Copy that, Nav. Thank you.
What is it? Is that a... What's underneath it?
Little polychaete.
Is that polychaete?
Seems to be nestled.
Watch
in
lead
home.
pilot.
Go ahead.
Thank you. We have seen this guy before, but
Okay.
that is pretty video.
Beautiful.
All right, come on up, video. All right, I'm moving on unless there's anything else you want to see here, watch lead.
This is great. Thank you.
Okay, thank you.
So we're seeing a lot of diversity here. I think we expected to see quite a few animals, and we are not disappointed. We're seeing some really interesting rocks and animals.
Yeah, so Shirley, you made a good point there. On the other geodive that we did a couple days ago, the animals were more sparse than this, but also the diversity was much lower. There was just the same thing over and over and over again, the same small set of animals, where so far on this dive, we're definitely seeing a much greater variety of sponges and corals both, as well as other things.
Okay, there's a fish back there.
Right, let's come in on the fish. I'm going to try to back up.
This one's moving pretty slowly. Probably gets
It's moving.
Yeah, it is moving.
It is moving.
And it gets
And
closer
it's
to,
moving down. And it's moving
which is
down.
not the
right
good
underneath me,
direction
just so you
for
know.
video. But at least we get a good view of the head. Head shape.
Yeah,
Come
so
on,
I think
guy.
this is a guy that we've seen plenty of times before. I'm pretty sure it's the Synaptobranchus dorsalis. I think we got a bunch of it just the other day. Very common eel down at these depths, voracious predator as well. So a lot of smaller animals, I think, are probably scared of this guy.
Thank you.
Great. Thank you very much. Well, our pilot and videographer have to be applauded for keeping up with this fish and
Nice job, Roland.
getting some, yeah, very nice job, Josh and Roland.
I got pushed back there. I'm going to push ahead a little
Yep,
bit.
thank you.
Ooh, rocks are much less, oh yeah, definitely much less covered with manganese, a lot of loose rocks. We are approaching the steeper part of the slope, and this is talus. Look at all the stalk sponges here, Shirley.
Dead ones.
Yep, dead ones, right.
It's definitely getting steeper.
Ah, shucks.
Do you want your uppers or lowers in?
They're good. I mean, I'm fine with them how they are unless we end up on a cliff.
Copy.
Just do a snap zoom on this. Closed anemone.
Looks like that might be
Cloud
an
of debris.
anemone.
Yeah, it looks like an anemone,
It is.
but it's hiding. Can't see much of it.
Is it eating? Is that coral in its mouth? What is that?
Oh, and it's got, what is, okay, what's happening with those branches there? They look like they may be attached. Is this, well, I was going to take a guess and say
anemones
stolen
eat
Everest, but I'm not going to
coral?
say that.
They,
Scott, what are you thinking about this?
Sure. Well, this sure looks to me like a hormathiid anemone, so that's a family of anemone, and it seems to be eating a chrysogorgid octocoral. How on earth it would have gotten it, I do not know, unless it's
Yes.
growing at the side of it and managed to grab onto some branch. It's a very
yeah, it's
odd
pulling it in.
observation. This kind of looks like the chrysogorgid we collected several dives ago.
Oh, check this out. Check out, wow, wow, wow.
That's what I thought.
Okay,
It's
pilots,
eating the
ships.
coral.
The question is how that happened, because
Yeah.
these anemones don't exactly walk around.
Nope, they
And
sure
neither,
don't.
They are.
and neither do the chrysogorgid. So you know what, Scott? I think your explanation is probably one of the more plausible ones. It may have been growing right next to it, kind of whipped next to it in this current, and the anemone latched onto it.
You would think there would be something left of the organism on the seafloor. It wouldn't rip the whole thing off.
Depends on how long it's been eating it.
But don't they have kind of a, they have a stalk that
Partial.
shows that they were attached? You'd think you'd see something on the
Maybe
rocks
so.
nearby.
Yeah. Well, that's a head scratcher, that's for sure.
Well, it's also possible that some other organism broke that piece of coral off and it kind of drifted by in the
current.
current.
Yeah, and I know what they're focusing on right now is trying to see also what's happening at that branch that's on the bottom, whether or not it shows an attachment point. But I don't think so. I think it's just another piece of this Chrysogorgia that's gotten caught in the tentacles. Interesting.
Yeah, this is weird. I think that was Tara.
Yeah, it was.
Made a good guess that maybe this has been a dislodged branch, and this hormethiid, when it can get some good food, is going to grab onto it. And you can see all the tentacles are retracted there.
Yep.
And it's holding on to this thing.
So, Scott, as we've been looking at this anemone, can you tell us how you would tell the difference between
That puts you at
the
9-0.
tube-making
Okay.
anemones and the ones that are not tube-making, and whether what group this is in? Oh, and as we're doing that pilot, wait, before you answer, since it looks like we're at a kind of good angle to come into this
And then if you can come
coral that's
starboard
right
to
underneath
center.
your lasers, can we get a look at the base of it, please?
Absolutely.
Although maybe...
Sure, Shirley. So that was not the tube anemone, which is in the group Theriantheria. That was a, quotes, true anemone, which is in the
Black
group
coral?
Actinia. So it had what we were just looking at had a very thick body wall, but it wasn't a tube. It was just thick tissue.
It's hidden
The
behind
tube
that.
anemones, you would see a
By-
tube. It had very
Probably
long,
rotate
slender
forward slightly just to keep it in view.
tentacles, and it would have a second row of tentacles in between the first ones. And so the way that was contracted, you wouldn't see it. So see, we're zoomed in on this black coral, the genus Bathypathes, and Tina Molodtsova from Russia, from the Shirshov Institute, has been very interested. She does a lot of work on black corals, and she's been very interested to see how many of these have associated with them this little scale worm, a little polychaete worm. And they often nestle themselves right on that main axis in between the tentacles of some of the polyps. And there may have been one there towards the upper third of the colony. It's
Yeah,
hard to tell.
right. Tina's saying that there might be a polychaete in the middle. Trying to see. Not sure we can see it from this angle here. Oh, they're right there. Maybe right there? Maybe right there, that darker mass.
Oh, yeah. Yep. Let me, I'll get a
Yeah,
better
we're going to
angle
just
on you.
move around. Yeah, is that what you were looking? Yeah, is that what you were looking at, Tina?
Yeah, I'm counting to 10. Yep, there you go. You found it.
Okay, we're just moving around.
That is,
Yeah, we're moving around to get a better angle on it.
Okay.
So, Pilot, how are you feeling about another move?
I would hold on for just a second now.
Can you bring down my port light again, Sean?
Yes.
Can't tell.
So indeed, that is a scale worm, which is a family of Polynoidae and polychaetes. And they are, polynoid scale worms are commonly associated with other invertebrates. It's not entirely clear what this one is doing with these black corals, if it's feeding on some of the mucus that it gives off or some of the food that the coral itself is able to capture. but I don't believe we've ever seen any instance where the worm is feeding on the coral itself. And I'll leave it to Tina to correct me if I'm wrong there. So thank you for this view. Very nice.
Yeah, thank you. So as we mentioned, these are community-driven dives. The expedition is science community-driven, and so we have many different interests on these dives. Each individual dive might have some different objectives. On this one, our primary objective is to kind of document the animals and the rocks. This might be an area that's marked for deep sea mining. In this particular case, we're seeing a lot of animals, a huge diversity of animals. Some of our scientists who are participating are really interested in some of the behaviors and associations of the animals to each other. So this gives us an opportunity to gather observations from which we can then generate some hypotheses to attest, to conduct further research. Beautiful crinoid here.
Good copy. Thank you, Bridge.
Oh, wow, that is gorgeous.
Very pretty.
And I'll just note that the crinoid is sitting on a bamboo coral.
I was waiting for you to say that. And they don't eat the coral, right? They're just using it as a place to attach onto.
That is correct. It's just a perch for this crinoid. The crinoid is trying to get its food out of the water column the same way that the coral is trying to get its food out of the water column. So the higher you get off the bottom, the better the currents, and so the more food that's delivered to you. So lots of these invertebrates are trying to climb up onto whatever they can find.
There you go.
We have a fish. A fish.
Now we finally see an example of this particular genus. I was hoping to see one of these.
And what is this genus? Don't keep us in suspense.
Well, Ken may bash this ID, but this is something called Porogatus. It's actually related to the cusk-eels. It is a cusk-eel, similar to some of these ones that don't have big eyes and big bulbous heads and things like that. This is just another variety in that family, which is very rich with a lot of different fish. So one of the things I'm looking at is the way he's moving his tail, and that's the only time I've seen that is with Porogatus identified in the northwest Hawaiian Islands, and it's quite a bit different and faster than a lot of the ophidiids.
All right, Pilot, that is me looking back at you now.
All right, so let's let him go.
That most excellent imagery is being brought to you by our team of ROV crew, our pilot, co-pilot, navigator, our videographer, Roland, and our video cutter, Annie. They are all from the Global Foundation for Ocean Exploration. Very important partner in this operation. They're responsible for maintaining and operating the vehicles, and also bringing you these just awesome video images that you're getting. Look at all of this diversity on this rock. We're seeing crinoids, at least 3 different species of sponges. 4, I take that back. Sea stars, octocorals. Wow. Wow.
It's right on the stem itself. It actually made it appear as though there was something.
Diva Aman, who was the biology science lead on leg 1, just said, ooh, indeed. Oh, this looks like E.T.
E.T. Right. E.T. came home.
This just shows you that all these suspension feeding things like to be on the upper parts of these surfaces.
All right. That's why Chris likes to live on the edge. Oh, my gosh, look at this. What the heck? Wow. See, now this looks to me more like a demo sponge, but it's not. Yeah, go ahead, Chris.
This is a bolosoma. We're actually looking at the backside. It's got a big, huge osculum on the other side that you can kind of see through the holes. It's quite open on the other side. We've imaged these a lot in the northwest Hawaiian Islands, and we finally collected one on the first leg of this year's cruise. So hopefully we'll be able to figure out what it is, and I think we're not going to figure out what it is. What we're going to wind up doing is giving it a name.
So we've had three. We've had E.T., we've had Pumpkinhead, and we've had Wally.
Wally.
Oh my gosh! Check this out.
So
it's like it's sticking its tongue out at us.
This is its scooper-upper of unconsidered plankton.
That sort of tells you where the prevailing current's coming from.
Yeah. Beautiful. Wow. So, Chris, this is facing into the current.
Right.
Oh my gosh! Check this out.
I'm not sure about this. It's possible that we're looking at his rear end right now, and his front end is the other side where water's entering. But I suspect that it doesn't even matter. Water may be coming in and out of it in different directions depending on the current.
Exactly.
And he's able to deal with it.
Because, in fact, what many of these sponges have are these in-current pores or ostia scattered all over their bodies, so they can get water coming in pretty much wherever the current is coming from, or wherever they can capture the particles. And then it goes through all of these canals and then empties into those larger canals that we saw so very clearly on this imagery.
Also, this is sort of a ridge line for Patty.
Yeah, I guess the way to sort of think about it is there's some similarities to our venous blood system where you're starting off with very small capillaries as you're draining the blood from the tissue, and they start to branch together or meld together into larger and larger veins until you get to the very large veins that actually bring it back to the heart. And so that's sort of one way to interpret water flow through these sponges. It's coming in through smaller holes, and then it's ending or getting expelled through larger holes.
So, Nav, how's the move coming?
We have 3 meters left.
Copy.
Just for info, in the first 40 minutes of our shift, we moved 120 meters, and that equates to just under 0.1. So we are okay.
Awesome.
But we're not
Copy.
making any time.
All right, pilot, I'm going to come down a little with you.
We will reach the end at this rate. We will, yes. But pilot wanted some leeway. So.
Yeah, try to get us an extra 15 minutes so we can sample if that's desired. So this, I actually had to go downhill to get to where I am now. I have rocks on forward and back of me. Zoom in on here real quick. Just something new.
Okay, so this is
Are we in a position for a quick zoom on that fuzzy colony in the back of the rock?
Fuzzy colony in the back of the rock.
On the top, there you go.
Yeah, I just want to be certain it's another Chrysogorgia
Sure looks
yellow
like it.
from a distance.
Hey, come in.
Yeah, Josh going in that.
Looks like the old one that we saw further down the slope, right?
Not sure.
Yeah, I don't mean the small one. There's one on the left.
Oh, okay.
Come on out.
Okay, we're going to look. Stand by.
Oh, yeah.
It's
on
right.
the
Oh, yeah, there's a big one. We see it.
Yeah, do you see? Is it the same thing?
I can't tell. But we'll go in and take a look. We just heard from Nav that if we continue at this same pace
Toe
that
is
we
bumping.
will make it to the end of our dive track. So we're doing pretty well. Okay,
Great.
now that, all right, see, so Scott, I would say this looks different, but I'm not sure.
It's just
Certainly
from
bigger.
a distance, it looked a bit more yellow in color, and I want to be sure we don't overlook any Okeanos bamboo corals, which have that yellow appearance.
We only have 10 seconds
But
here, and
this
then
is indeed another Chrysogorgia. You can see the beautiful golden color of the skeleton. So Chryso translates as gold, so the golden corals, and here's a perfect example of it. And what's also cool about what you're looking at is that you can see right through the tissue to that gold-colored skeleton. So there is tissue on there. All of those little polyps are arising from tissue, but it's so thin and transparent you can see through it, even though all of those polyps have their digestive tract connected by small tubes running through that tissue. And then there was a squat lobster in there. Thank you for that zoom.
That was pretty.
Take a look at the view from Sirius down on
Copy that.
D2.
All the way up.
I think that's on feed 2 for those of you who are watching, listening from home. We are on the NOAA ship Okeanos Explorer, and we are coming to you live from a place called Boat Gio on in the Mariana Trench National Marine Monument. This expedition is actually part of a 3-year campaign called Capstone. Capstone stands for the Campaign to Address the Pacific Monument Science, Technology, and Ocean Needs. And it was designed by NOAA. Oh, look at that beautiful sponge. To address the needs of our nation's resource managers for these monuments, to better understand the deepest parts of the monuments. Yeah, go ahead.
Just like the one
It
that
does
we
look
saw
like the one
at
that
about
Chris thought might be
1939. Yeah.
that Chris thought might be
a
something
new sponge.
Well, anyone want to consider a collection? Is that a possibility? I'm not biased at all toward collecting sponges,
Of course
am I?
not. No.
Okay,
And
so
this is a new one.
Copy that, Nav.
What do you think?
New, we've not seen this before, Chris.
Yeah, I think that may have been the first time Chris has suggested a collection of a sponge.
Really?
That's fine. You guys are the sponge experts, and this also looks different to me, so it seems like a useful collection to me.
If you want to ramp up my
Okay, and we
Should we
have
look at
about...
the attachment?
We can look at the attachment
Draw
first.
us
Rest of our biologists, what do we think? Yay, nay? Remember, we have about 4.5 more hours? 4.5 more hours.
And Tara Luke says it has a tiny crinoid on it.
Do you want that
a little bit.
report swinger, man?
Tina also notes the
See
crinoid.
the root or the base.
What does the attachment look like?
Go ahead.
Oh, yeah, I think we should get the rock, too.
Yeah, copy.
Don't you? You want that attachment.
You mean that rock that's in place? That rock?
Sure.
Yeah. All right. Got it.
All right, pilot.
Got it. Okay, so as long as we can get some good imagery of the base before we
Do you care
collect
if
it,
I
because
rotate
it
the
is
port towards you?
quite
There's nothing on
unlikely
this
that we
side
will
of
be
me.
able to get it when
My
it
sonar, and I should
comes up
move
off
that
of the
much.
bottom.
Copy that.
Yeah, it might pop off.
And then
Never
I can
know.
switch cameras with some setup.
Yeah, so that's a nice image because no, you won't be able to get the base. I mean, you can clip it somewhere down on the stock if you're going to collect it. So we've got information on the base as a result of this close-up, so that's great.
Okay, great.
Chris, is this one continuous stalk, or is this one growing off another stalk? I can't tell that fuzziness if that's spun sclerite or something else.
I think something's growing on it, and I think this is one continuous stalk, but not sure.
Yeah, so I asked the question just because I know you say there's that important difference between, I forget what you call it, but the the spicule anchor versus the hold fast.
Satisfied.
All right, come on out wide. All right, I'm going to set up now.
So the terms, I don't think anyone's probably going to want to memorize this except for me, but we call a basiphytus attachment, where it actually creates a plate, like what we were just looking at, a plate that glues onto the rock. And then if it anchors itself via loose spicules that hook into the rock, we refer to that as a lophophytus attachment.
Eventually, I will learn that, Fritz.
Hydraulics up?
Yes, hydraulics are up and you're ready to go.
So while we're setting up for this collection, you can watch as this sponge is going to be collected. Just wanted to let everyone know about this mission.
Just want to point
yeah, go ahead, Chris.
out.
Yep.
He can grab that stock and it should remain fine. I mean, it'll cut pretty easily. And this is a soft sponge. It shouldn't be brittle. It should be quite an easy collection just to grab it by the stalk.
Yeah, copy that.
Okay, thank you, Chris. Pilot copied. This campaign, this mission was planned and, broke right off. It's being conducted by the NOAA's Office of Ocean Exploration and Research. We have other NOAA partners, the National Marine Fisheries Service, the Marine National Monuments Program, the Deep Sea Coral Research and Technology Program, and the National Center for Environmental Information. And that last partnership with NCEI is what's making it possible for us to be able to make these data and images available to you, to the public, to our scientific colleagues, the scientific community, in really near record time. It's takes about 2 to 3 months to make all of the data available. But many of you, I'm sure, have been checking the website, the Ocean Explorer website, have been able to see images and video clips posted sometimes the same day or the very next day. So this is really different from the way we typically operate oceanographic expeditions. Oh wow, look at that image. Very cool. You can really see down into these atrial cavities of this glass sponge.
That drawer's open.
So the dives,
Yeah.
once we...
one other thing to be aware of is this guy might float. So before he releases it, if he could get it down as far into that box as possible, that would be helpful.
Yeah, copy that.
Okay, thank you.
I was just realizing that.
Thank you.
So we might...
Do we do any upward thrust?
We might... Let's see how stable it is.
This pilot's considering what to do. Okay, we're putting this in. I don't know if you can see this. I think you should be able to have all these screens up. Okay, that's okay, Josh. What we might end up doing then.
Close.
Yeah, close. Good.
Got it.
All right. So the stem might be a little caught on there, but we definitely got it.
Yep, that's good. We got it. And I think maybe if we collect anything else that's going to go into a bio box, we'll put it on the starboard side.
Oh, definitely.
Okay, great. Thank you very much. That was the second sample.
Floaters, always fun.
All right, stand by, those of you on shore and at home, while I record this. This is
I'm going to have you
port inboard
put it in the
bio
box, and you
box.
can close it on the stem.
Okay.
So
Yeah.
I'm just going to hold it in the
It's up-
box.
yeah.
we'll try to close it on the stem.
It's not a bad idea because then it might not even break the stem if it's
Yeah,
flexible.
it'll still seal the box.
Yeah.
So. Another holothorian.
Yeah.
Just do a quick zoom for ID.
How do we feel about a move, gents?
I think we're good for a move.
Yeah, we just need to rotate back, pilot.
Oh, okay, that's right.
I'm looking at about 90. So we need to rotate 30 degrees port after this.
30 degrees.
Oh, I'm sorry, starboard. 30 degrees starboard.
Okay, so there may be a candelabra. Oh, I think we may have passed it. Sorry. Oh no, there it is.
To the
On the
lower
far right and the lower
Push
right
you
too.
off.
Yeah, far right. Let's take a look at the branching. I think that's a candelabra.
This guy just right
Yeah,
under the lasers.
right underneath the lasers.
Okay, come on in video.
Loaded with brittle stars, wrapping their arms around the branches of this coral.
Yeah, this looks like another primnoid octocoral, an octocoral
Okay.
in the family Primnoidae, possibly Candidella. And at least in the North Atlantic, there's a species of Candidella that we see that often has polychaete worms associated with them, such
Yeah,
that
Nav.
the presence
Pilot's
of the
good. I think we're
worms
good.
causes
Put
the
a move in now.
coral to
I'm back
change its
towards
growth
120
pattern such
and have
that
a
it
view of
creates
pilot.
essentially little tunnels or garages that
Bridge, this is Nav.
that the worms live in, and there may be hundreds of these worms on a single colony. And I can see that some of the branches of this colony look thickened, and I believe that's where these polychaetes would be living
Okay.
if they're there. I'll bet they are there.
Yeah,
And
there's
it's also very common to see all these brittle stars on those particular species.
Yeah, and Scott, Candidella helminthopora, which is named after that the species that has the worm in it, has been documented in the Central Pacific in Hawaii.
Alright, let's come out. Yep.
Can we get
Bridge,
close, husband?
this is Nav. Hello, Bridge. Could I have SEM again, please? 30 meters, bearing 120, speed 0.2 knots.
Video clear?
Video's clear.
Okay.
Good copy, Bridge. Thank you.
Alright, how's that looking, Sean? You centered up?
Yeah, that looks perfect.
Okay.
Another couple of Walteria sponges, maybe even 3.
Let's just do a partial here.
Yep,
Okay, hold
another
that.
Walteria. We've seen quite a few of these. These have been quite common. That one in the back that has kind of a flared top.
Is that of interest?
We'll do a snap zoom. We'll just zoom in real quickly on that, please.
Come on in.
Beautiful. Thanks, pilot.
Okay, can you clear video?
Okay, so we're seeing some more black corals, a couple different species, Bathypathes and Atricipathes. Deb Glickson is commenting that there are some angular parts of this outcrop. And we're seeing again some more of these dead sponge stalks.
It's coming partial here. Stop.
Beautiful stalk crinoid there, I believe. Yep.
Yeah, I think the current
Again, we're seeing
has our tether
a few
pretty
branched corals and some lyrate or candelabra-shaped corals that could be different as well. Okay, so this is another one of these, wow, looks like a weird Walteria that's got, looks like brittle stars on it, a couple different kinds. Over to the left, another whip coral. And even further left, looks like a younger Walteria sponge, hexactinellid sponge.
Okay, we can come out. Come on in. It's a stock. Alright, come on up.
Okay. So certainly seeing a lot of sponges and corals as we expected that we might find here. So it's always nice when we can go to a spot that we predicted might be very diverse, have a lot of fauna, sponges. We've seen corals, we've seen polychaetes, we've seen holothurians, barnacles, other crustaceans. Really, really cool. Something branching in the distance. Looks like it's got it's behind the black coral. Yeah, you're heading towards it. It's over to your right. Oh, that might just be, sorry, that might just be the rock that's looking like there's branches on it. Okay, so there's a black coral to the lower right. We're heading straight for another whip coral. And can we get the nav quad back, please? Thanks. So typically what we do when we're making a collection is switch over the quad view screen that you can see so that you can get see 4 different camera views so you can watch the entire collection. And we get so wrapped up in what's next that we're looking at that we
finally put another move in now?
Yep.
You're still pretty good.
Appreciate your reminders to get the other quad screen back up on
You got
feed 3, I believe it is. Another one of these are big hexactinellid sponges that we're seeing here. This one, Chris, this one on the, these roundish ones, we've seen quite a few of these now.
Fine with your sides?
Yeah, so Shirley, this is something that's in the Hyalonematidae, and we actually collected 2 of these last year.
Okay.
It's Hyalonematidae subgenus Hyalonema subgenus Corinonema. And
did you
so
copy
we're going
about
to find
the,
out presumably
I'll put
what
another move in now
species
if you
these
want.
are, but they're very distinctive sponges,
yes, if
and
that puts us on course
underneath
still.
them they have the fibrous attachment. You just can't see it.
Right, so
But
hyonemas usually...
collecting one of these revealed that it was a hyonemid.
Yep, and we've actually seen quite a few of these over the last few tens of meters of this dive track. And yes, as you point out, the hyonemas usually would have stalks, and it's really hard to see those stalks from these really squat, rounded, kind of like flattened spheres. So very interesting observation. From your prior collections. We have a long-stalked hexactinellid sponge, another anemone, something in the foreground that might be a chrysogorgid, but I'm not going to say for sure because some of these have not been. You want to write, yeah, quick zoom on that. This looks like it's a chrysogorgid. It's got I think a squat lobster in there. And I think as Scott France pointed out earlier, in some of these you can actually see their golden skeleton underneath, the golden stalk. This is why they're named Chrysogorgia. Chryso means gold. And we've had confirmation from Scott, from Tina Molitsova in Moscow, and from Asako Matsumoto in Japan, that yes, indeed, it is a chrysogorgid.
Are we clear?
Thank you, pilot.
Come on up. Moving on.
We are at 1,850 meters along this dive track. A place called Vogt-Gio, V-O-G-T, Vogt-Gio.